So today I will be talking about a work that we're doing here at the Stanford Genome Technology Center, and it deals with looking at a group of white blood cells called neutrophils and their behaviors in ME/CFS. So I'd like to start off first by explaining what we think is causing ME/CFS. And what's possibly causing ME/CFS is the body's inability to properly turn off inflammation and heal. Bob mentioned some things about this earlier this morning, so it might be familiar. So normally when a patient or a healthy person faces an infection or an injury, the body triggers inflammation to fight off that threat. Once the threat is gone, the body activates critical cell and biochemical events that resolve the inflammation, and it initiates healing and returns the body back to a normal state. But in people with ME/CFS, this process gets stuck. They start the inflammation, but it doesn't get properly shut down. And it causes ongoing chronic inflammation. And this leads to a lot of dysfunctional immune system processes. So what we're trying to do to help is that we're looking for chronic inflammation markers. And specifically, we're looking at neutrophil behaviors. And we hope that these markers can provide evidence of illness for patients. So you may be wondering why we're looking at neutrophils. Well, neutrophils are the most abundant white blood cell. They make about 60% of all your white blood cells. They're highly sensitive to any inflammatory signals, and they respond to inflammation. They're the first responders to any inflammation event that you have. So if something goes wrong with them, it's likely to throw off the immune response process. So a little roadmap of what I will be covering today. My hope is that by the end of this presentation, you will know why we're studying neutrophils in ME/CFS, and then what they can tell us about chronic inflammation. And I'll talk a little bit more about that later. What neutrophil behavior we're actually looking in patients, and then how we're performing these tests. I will also like to share about what neutrophil behavior we're observing in ME/CFS patients. And finally, I'll conclude with what we think is causing these neutrophil behaviors in ME/CFS patients. So the main question that we've had and we wanted to answer is ME/CFS a chronic inflammation disease? And the reason for this is that if you look at an ME/CFS patient and you compare it with a chronic inflammation patient such as MS or lupus, you can see that there's a lot of significant overlap in their symptoms. But when you take an ME/CFS patient and a healthy control and use these typical blood tests used for inflammation, ME/CFS patients really don't show strong signs of inflammation. And that's because these tests that are used in the clinic, they only detect active inflammation. They do a very poor job at detecting long-term chronic inflammation, which is what we think is involved in ME/CFS. So to know how to test for chronic inflammation, it's good to learn a little bit more about the inflammation process, and I'd like to walk you through that today. So if you think of your body like a house, when something goes wrong, such as a bacterial infection, as you can see here in the schematic, your body calls the pest control team. This pest control team are the neutrophils, and neutrophils act like little tiny warriors that migrate into the site of the tissue, and they use different tools to fight off the invaders. Some of this include like reactive oxygen species, where they release toxic chemicals to break down the walls of bacteria. Or they can also shoot out sticky webs called nets or neutrophil extracellular traps, where they act like little mini Spider-Mans that trap the bacteria in these web-like structures. But this is a suicide mission. They usually die whenever they excrete their neutrophil extracellular traps. Or they can even gobble up the bacteria like little Pac-Man, and this is called phagocytosis. So if everything goes well and they're able to clear up the bacteria, then they're able to move on to a cleanup and repair phase. And this is what's called inflammation resolution. In this phase, those neutrophils, the ones that fought the battle, are no longer needed. So they need to undergo a programmed cell death called apoptosis. And then another cell shows up called the macrophage. And these are kind of like the cleaning crew. They ingest the apoptotic neutrophils, and this helps the tissue heal and return back to normal. But there's a problem. Sometimes the bacteria aren't properly cleared out, or neutrophils just don't die the way that they're supposed to. And when this happens, the battle drags on, and we enter a state of chronic inflammation. So, what studies have shown us is that when there is chronic inflammation, several things go wrong. There's probably too many or too little neutrophils coming into the tissue. Sometimes they don't die properly, so you get abnormal apoptosis. And they keep releasing the toxic chemicals and webs that they use to defend themselves, making the tissue, damaging the tissue where they're at. And instead of healing, some cells die violently by breaking down their skin or membrane, which is called necrosis or necroptosis. And this causes even more inflammation. So, understanding these markers are critical because it allows us to select the ones that we actually want to test in patients. So, going over that, we wanted to test if neutrophil cell death was different in MECF patients versus healthy controls. We wanted to determine if they were dying through apoptosis, which tends to be non-inflammatory, or were they dying through deaths such as netosis or necrosis, which are a lot more inflammatory. We also wanted to test neutrophil ROS production. And finally, we wanted to look at how neutrophils were migrating. We also wanted to understand how these neutrophils in patients deal with an inflammation threat. So what we did is that when we acquired the neutrophils, we would then treat them with an inflammation agent. So I'll walk you through a little bit on how we performed these tests. So we were able to collect blood samples with the collaboration of Michelle James. She'll be speaking next. And we collected from MECFS patients and healthy controls. We took the blood sample and isolated neutrophils from them. We took our neutrophils, and then we used different stains that will allow us to determine if the cell is undergoing cell death, if they're producing ROS, or if they're undergoing apoptosis. At this point, we also activated them or treated them with that inflammation agent. We placed the cells in a live cell microscope where we would then image them for every 90 seconds for a total of 3 hours. From this, we were able to gather about 1,000 images per patient. And this allowed us to produce data trends of neutrophil behaviors of the patients. So now that you know how we did it, I can share what the patient data is telling us. So the first question we wanted to ask was, do more MECFS neutrophils die? And here you can see to your left, we have a healthy patient, and then to your right, you have an MECFS patient. So from this, we can count the cells that are turning green, which is an indication that they're dying and that their exterior skin or membrane is breaking down. And we can plot the dead neutrophil population percentage over time. And we looked at 10 MECFS patients and 10 healthy controls, where MECFS patients are shown in red, and then the blue shows the healthy controls. And what you can see from these plots is that MECFS neutrophils die, a lot more of them die, and they die even faster than healthy control and neutrophils. While healthy control neutrophils tend to survive a lot better, especially during early on of our tests. And what this is telling us is that MECFS neutrophils may be more fragile and they die easier, which can contribute to an inflammation state. Second, we wanted to know about apoptosis. In particular, we wanted to know if ME/CFS neutrophils had abnormal apoptosis. So here we collected the images for this. We stained the cells, and those cells that are turning turquoise are those that are undergoing apoptosis. We then counted them, and we saw that ME/CFS neutrophils showed less number of neutrophils undergoing apoptosis than healthy ones. And what this suggests is that ME/CFS neutrophils don't die properly when they should. And this can prevent the body from resolving inflammation. Third, we looked at if ME/CFS neutrophils produce more ROS. So ROS, again, is that toxic chemical that neutrophils use to kill the bad guys, but they can also damage healthy tissue if it's produced in excess. So you can see from our videos here, the ROS indication, we were able to quantify the number of neutrophils generating ROS. And from the data plot to the right, you can see that there's more ME/CFS neutrophils that make ROS. If this is a case in your body, then it might cause more tissue damage and worsen inflammation. And finally, we looked at how neutrophils were moving. So for this, we acquired images of the cells, and using a software, we tracked how fast and how far neutrophils moved. From our data, if you look at the plots at your right-hand side, we captured frame-to-frame speed versus time for ME/CFS patients and healthy controls. We also captured frame-to-frame distance over time for this patient population. And what we can see is that ME/CFS neutrophils are traveling at lower speeds than healthy controls from this plot. However, and they also travel less distances than healthy controls. However, the difference is only significant for frame-to-frame speed. So what this indicates is that ME/CFS neutrophils migrate at a slower speed than healthy control neutrophils. And this may be causing them to take more time to reach inflammation sites. So what our entire analysis is bringing into focus is that chronic inflammation markers based on neutrophil behavior, as shown here, do show a difference between ME/CFS patients and healthy controls. If we continue to see these trends in more patients, and these markers or set of markers are unique in ME/CFS patients, are unique only for ME/CFS patients, then these can be used for a diagnostic panel. And finally, I would like to share what I think or what we think is causing these neutrophil behaviors. And similar to Bob Navio, we also think that there is a malfunction in purinergic signaling. And the way that this works is that if you have neutrophils, when they're exposed to an inflammatory event or if they die, they produce a chemical called ATP. And ATP behaves like a fire alarm. It is released usually when cells are damaged or they die or are triggered. And ATP binds to these receptors found on the surface of neutrophils, as you can see here. These receptors behave like our ears. When ATP binds to the receptors, they guide their behavior. And it's the same way if we were to hear a fire alarm, it would guide us to leave the building or to put out the fire. But what happens in ME/CFS is that these receptors may be overly available or overactive. And what this leads to, it's poor cell death timing, or increased ROS, or impaired neutrophil movement. So a good way to see it is it's like if we were to grow 8 more ears and have a lot more fire alarms going off, we wouldn't be able to respond properly. We would be too overwhelmed. So What we believe is that there is a breakdown in this purinergic signaling in ME/CFS patient cells that may be preventing their bodies from shutting down and resolving inflammation properly. So, as I mentioned in the beginning of my presentation, my hope was that by the end you would know what neutrophil behavior we're testing and how. So we're looking at neutrophil cell death, ROS, and migration. And we're looking at behavior trends. We want to see how they behave dynamically. Second, we wanted to inform you about what we're learning about neutrophil behavior in ME/CFS patients and what it could tell us. And what we're seeing is that ME/CFS neutrophils have abnormal deaths, ROS, and migration, which point to chronic inflammation. And what is possibly causing this neutrophil behavior in ME/CFS? Well, we think that it's malfunctioning of purinergic signaling that doesn't allow the body to heal properly, similar as to what Bob mentioned earlier. So what we plan to do next is we want to expand our cohort. So we need more patients to test this on. We also want to explore the purinergic receptor signaling in ME/CFS. We want to look at how much ME/CFS neutrophils are producing ATP, and then what's the receptor expression. And finally, if we do identify receptors that are normal in ME/CFS patients, are there therapeutic targets that would allow it to that would allow the restoration of inflammation resolution? So with that, I would like to end my presentation here, and I would like to acknowledge those that contributed. Obviously, Ron, who's been a mentor here at the Stanford Genome Technology Center. John Michael as well has been another mentor. And then Michelle James, who's been the reason why we've even gotten these blood samples. So we've been collaborating and seeing how we can merge our data together to make it more powerful. And a special thanks to all the patient participants. We know how much of a sacrifice it is to come in and donate, so we really appreciate it, as well as the Open Medicine Foundation, who's funded most of this research. And finally, I would like just to end with a call to action. We really want to encourage the participants to please learn about our studies, and if they feel comfortable, to please participate. The reason for this is that we do need more participants in order to make our studies significant. It's the only way that we'll be able to be accepted among the broad medical community. So with that, I'd like to thank everyone for their time and end the presentation here. Thank you.